Okay, welcome everyone to the first day of Research Dialogues. My name is Lisa Piazza and I am the Director for Undergraduate Research. And I'm so excited to welcome all of you to this session, our moderators, our presenter panelists, and certainly our attendees. We have had a wonderful, exciting day full of just great conversations, panelists, and ideas and topics. And as you can see, I'm really excited because this is my favorite time of year. I help students all year long with undergraduate research and getting into research, and this is a real treat to see the actual presentations that result from all of that hard work. So welcome everyone. I want to turn it over to our moderators who will lead the session today. If you are attending this session, we certainly invite lots of questions. And so at any point in time, please go ahead and put your questions in the Q&A tool and we'll get going.
Well, welcome to this Q&A panel. I'm Rachel Fleming. I'm Scholarly Communications Librarian at the UTC Library. And I work on a whole range of issues from affordable education and the sharing of information through traditional and open publishing. I'll be one of your moderators, and I'm joined by Dr. Kristen Black, who will be our other moderator. Do you want to introduce yourself real quickly?
Yeah, sure. I'm Kristen Black. I'm a member of the faculty in the psychology department here at UTC. I've been here about 4 years now, and I've gotten to experience a lot of different research dialogues with in-person and virtual this year. Most of my research is in the area of workplace health and well-being, so that's where I fall in the research spectrum, but I'm excited to get to hear from a really diverse set of presentations today from a lot of different areas. So Rachel's going to introduce our panelists next as we get started.
Yes, we'll just have our panelists introduce themselves, and I'll call on you to keep it going, starting with Elizabeth.
Hey guys, I'm Elizabeth. I'm a senior at UTC this year, and I did my research with Dr. Giles. Is there anything else I need to cover?
No, that will do. Next up is Taya and Juliet.
Hi, my name is Taya de Blonk.
Hi, I'm Juliet Wimberly. I'm a pre-professional biology major with a minor in chemistry. And we are doing research with Dr. Alda on the minnow Campostoma.
Yeah, we used PCR amplification methods to get sequences of the cytochrome B gene, and also used ArcGIS for our project. So we're excited to answer some questions if you guys have any.
And next up is Vaidehi.
My name is Vaidehi, and I'm a junior at CSAS.
Katherine.
Hi, everybody. I'm Katherine. I go by Katie most of the time. And I am a sophomore psych student here at UTC, and I'm also in the URTOPS program. And I have done my research under Dr. Ashleigh Pipes, who is also here today. So I'm in psychology, she's in linguistics, and our project is on the intersection of creativity and linguistics and language learning. So it's super interesting kind of interdisciplinary work there.
And Dr. Pipes.
Hi, I'm Ashleigh Pipes, and I teach in the English department, but study linguistics. So the particular project that we're working on, sort of parallel things, as Katie mentioned, is the relationship between a person's creativity and how well they speak in a foreign language. And we found that it does indeed have a relationship, and but more so when people are speaking to another person, not when they're speaking alone. So, so thanks so much.
Austin.
My name's Austin, and my co-researcher Jacob Smith is on camera too. I am a major in environmental science with a minor in anthropology, and he is a anthropology major. We conducted our research with Dr. Morgan Smith, reassessing historic accounts at the Battle of Chickamauga out on the Chickamauga Chattanooga National Military Park.
Jacob, anything to add?
No, ma'am. I'm just a sophomore myself, so I think that's the only thing that was missed.
Dr. Persons?
Hey everybody, I'm the director of the Jeffrey L. Brown Institute of Archaeology here on UTC's campus. If you're interested, come by Brock Hall. We're in the basement all the time. So my research is presenting on some work in the Virgin Islands. That's kind of an ongoing project. So I'd like to commend everybody. Y'all did a great job on these presentations.
And finally, Taylor.
Hi, everyone. My name's Taylor, and I'm a sophomore here at UTC. I did my work under Dr. Smith as well in the archaeology department here on campus, and we're working on digitizing old documents that are housed in the institute on campus.
Great. And I encourage everyone who's here who hasn't had the chance to take a look at the posters and presentation that these panelists have done to go ahead and do that after the session. They're all really excellent. And I'm going to throw it to Kristen to get us started with the questions.
Great. Thanks, Rachel, and thanks for introducing everyone. So as a reminder, if you want to use the Q&A box, if you have questions for each other about specific projects or about research in general, you can use the Q&A. But we wanted to start with a question, knowing that in most all of our research, things don't always go as planned or end up being as easy or straightforward as you expected. So I wanted you guys to share a little bit about the biggest challenge you've encountered while you were completing your specific project.
I'll go ahead and start, I suppose. Jacob and I were working in ArcGIS Pro, basically georeferencing historic accounts against known artifact positions from a recent archaeological survey done at the national park. Basically, our biggest hurdle in completing data treatment for this presentation was ArcGIS programs in itself, a lot of formatting issues back and forth. And once we got there, we found that some of the stuff was out of whack. So it was just a constant back and forth of georeferencing points and getting them in the correct location as we went through our data.
I can go next. So for my project, I was trying to create a vegan meal replacement that was still high in protein, so at least 25 grams of protein, since vegans have a hard time getting protein in. And one of the biggest challenges was keeping the calorie count low, because whenever I had high protein, I would have like 700 to 800 calories in a 4-ounce jar or in like a small protein bar. And then if I did lower the calories, then the protein would go down again, because protein has about it's either 4 or 9 grams or 9 calories per gram. So that was a big challenge. And then also that affected the flavor as well. So I had to find ingredients that were high in protein, low in calories, and then also tasted good and had a long shelf life.
Elizabeth.
Okay, so for my project, I think our biggest problem, so we worked with piscidins, which we got from our collaborator, Miriam Cotton at another university. And with COVID, she had trouble, having enough piscidin to sustain our research. So there was a bit of time where we were kind of just verifying everything we had, because that's all that we could afford to do. We didn't have enough of the peptide that we needed, but eventually we were able to get more and finish up with everything that we had planned on doing.
Katherine?
Sure, yeah. COVID also presented some interesting challenges for us, especially because we're doing people-based research. So originally, brainstorming kind of this project, I had some ideas about maybe we would be able to go into a classroom and work with, students there. And we ended up opting to do everything virtually, just because that's way more practical right now. But one of the things I didn't necessarily expect at the beginning was how much detail goes into creating all of those different pieces, like all those little artifacts. So getting our surveys together, I put everything in a Google site just to get it all centralized. And then it kind of became this project, and a kind of sub-project of user interface and trying to make things actually work for participants, and where do I put the informed consent to make sure that people actually see it, rather than having to click off 3 pages or something, which took it made everything take like 4 times as long as I originally anticipated. But Dr. Pipes, you might have some interesting notes to add just on how your book kind of plays into that, and how looking at the overview of the project and doing an undergrad project, if you want to chime in.
Certainly. And one of Katie's challenges this semester was to see if a project could be done start to finish in one semester, because that kind of goes along with one of the chapters in a book I'm working on. So she's a bit of a guinea pig there and has had to face all the challenges of it. And it's interesting too that the challenge for her independent project has been the interfacing with participants virtually, because before she spoke, I was thinking my greatest challenge in the original project was with the participants in person, because I was quite fortunate that I had a lot of willing participants, but they were all available just in one short 2-hour block. So usually it's hard to find participants, but I had plenty of participants and ended up training undergraduate research assistants to be extra data collectors. So it all kind of, no matter which way you go about it, online or in person, I guess there are equal challenges.
I was really interested to hear that there were some troubles because of COVID, not just in this interpersonal interaction, but with a more scientific paper. Did anybody else run into anything during your research process that you didn't expect because of what happened with COVID?
I don't think that this is maybe directly related, but I actually work at a hospital. So of course, I'm sure, as you can imagine, that got a little complicated. I actually ended up having to take some time off of work because it was best for both my director, me, and everybody else who was also working in the lab. So that was something I didn't expect to have to do. It's a small town hospital, and up until just a few months ago, really, COVID hadn't hit my town. So that was very unexpected.
It's really helpful. I think there's a lot of interesting themes that everyone's original plan didn't always go as expected, whether you had to change data administration or having difficulty with equipment and having to adjust. So that seems to be a common theme that we all experienced. I know Brooke or Taylor or Taya, did you guys have anything to add for that question? If I missed anyone else.
Yeah, for the biggest challenge that we encountered during working on this project. So for our project where we're working with DNA, specifically mitochondrial DNA, it's oftentimes a very precise and time-consuming process, and it can be kind of disappointing whenever you spend hours in the lab working on a reaction for it to not work whenever you get to the stage where you're putting it into the gel. So that's kind of disappointing, but it does make it even more rewarding whenever we work hard and finally get a perfect gel or the results we want to see after a lot of trial and error.
Can I follow up and ask you what would make it not work?
There's different parameters for a reaction. So it can depend on the amount of magnesium you put in to the reaction you did. It can depend on the quality of the way you injected the DNA into your gel. So it's a lot of different factors, if you have anything to add.
There could also be any type of contamination, like any dust mites, anything in the air,
Right.
Temperature fluctuations. Some of the samples were quite old, so it depends on how well preserved their DNA actually is. So there are a lot of challenges, but we were able to mostly work around them for the most part.
Yeah, we ended our project with about 1,047 samples in our tree. So yeah, it worked out pretty well in the end.
Thanks. Well, I can say that one challenging thing here in the archaeology lab is that, usually you're working as kind of a close part of a team, whether you're in the field or in the lab. And so We had to kind of socially distance our lab, like the rest of the labs on campus, to make sure that people are performing research in a safe space. And it's just certainly been challenging. I know Austin and Jacob, you guys probably have something to add to that about working in the field during COVID, which is usually, again, a very collaborative, kind of close contact sport.
It really couldn't have worked out any better for Austin and I. And well, really the whole ANT 3350 class, due to the fact that, we had plenty of room to social distance, working in well over, 20 plus acres of land, surveying during the time. So it worked out very well for us. But in the lab, like I agree with Ms. Pearson's, it was tough at times.
I think a lot of the problems I ran into are very technical. I was working with a book and document camera scanner. Well, I originally started with just a regular old copy scanner, and it turned out to be very time consuming to do it that way. So Dr. Smith ordered just a regular book camera, and we scanned things that way. But it could be a little frustrating when you would spend a few hours scanning things and then the files would be corrupted. So the next day you'd come back and have to just scan again. So definitely working through those technical issues and being able to bounce back the next day was probably my biggest challenge.
Learn a lot of personal resilience in conducting any involved research project too, right? Well, thank you all for your perspectives. I'll let Rachel lead us in our next question.
So what was most surprising for you working in the field that you were working in? What didn't you expect to run into in your research? Let's start with Austin.
I think one thing that both Jacob and I didn't expect was more on a single item basis. There were a few artifacts that kind of came out of the woodwork as we were going through and identifications that really brought some different overall, outside of the Battle of Chickamauga, the context of every operation that happened on the military training field, as it were, for the time. Just finding different modern things, historic artifacts that really just kind of could either paint a picture or present something that we weren't really aware of being around in that time. And I'm sure Jacob has something he wants to add on that. So many artifacts we've looked at.
Like Austin said, there were so many outliers to what we did find. We had to really zero in what we were going to, discuss on this project due to the well, in the very beginning, one of the young ladies found a 11,000-year-old point, and that was it. As interesting as it is, it had nothing to do with the Civil War itself, nor did any of the 30-06 casings that we found laying throughout the entire field itself at the Brotherton cabin site that were from World War I and World War II. It's the outliers it really did make it difficult in that sense as well.
Maybe let's go to Dr. Pipes.
Sure. I was just thinking, as far as unexpected, for me, it would be cultural. Again, I mentioned all the participants. I was conducting my original research in Turkey. And so I went in with an American mindset of we'll schedule everybody in these neat little 10-minute blocks. And I had a sign-up sheet and a spreadsheet, but that's just not really the approach that was common where I was in Ankara, Turkey. It was more like everybody was happy to stand out in the hall and wait their turn and just cycle through. So I was having a little bit of dissonance between the way I would have expected it to be done. And I would have thought from an American mindset, if you're having to wait in the hall for an hour for your turn for a research project, you might be pretty mad, but nobody was. They just went and got a cup of tea and came and waited and seemed happy as could be to participate. So that was unexpected for me.
Katherine?
I I found kind of, let's see, I'm trying to figure out how to phrase this. It was so interesting to me because it was both unexpected and I didn't expect it to be unexpected, if that makes any sense. But one thing that Dr. Pipes has been great about encouraging me to do, and she's had to bring it up several times just because it doesn't occur super naturally to me, but the benefit of just doing something, like just engaging in a piece of the project. So like, you know, the part about sitting and brainstorming, like, what are we going to do, and putting all those details down on paper and then going in and working on like the surveys and stuff, those things were kind of flowing fairly naturally. But then sometimes I'd get stuck on something and she'd be like, I think you need to just sit down and like just sit for 2 hours and work through the Duolingo portion, because we're doing like we have participants actually learn some Turkish, and it's about an hour and a half to 2 hours of Duolingo. And once I was able to sit down and engage firsthand myself with that piece of information that I was expecting from participants, I had all of these new insights that came immediately that I had sat for hours and not been able to think of. But I was just in a conversation with my mom about laughing about the difference in Turkish. I think the word for thank you is like really, really long. And it was only, at least in our initial set of like 75 words, it was only giving one option for thank you. And I was like, we have a million ways to just kind of quickly say thanks or appreciate it or whatever. And because I'd had that interaction with my mom where I'd actually been able to utilize that new word and talk about it, I remembered that one way better than the rest of my words, which ended up being something that fit right into what we were thinking about research-wise with, you know, how does deconstructing words as you go and that sort of creative piece of how would I use this in real life impact actual language retention and use later. So I wasn't necessarily even looking for an insight there, but it ended up being one of the more valuable pieces. Just came from just sitting down and doing it myself for a minute. So definitely good advice from Dr. Pipes. If you ever get stuck, just do it.
And Dr. Persons, your poster is about like a step in a larger research project. When you look at all your papers, it's like this happened and then this happened and then this happened. Like, does it really go like that in your process? Or
it's pretty, it feels like it's never ending right now. In this case, we excavated what is comparatively a very small archaeological test unit that's really only 2 meters by 50 centimeters wide, which again is extremely small amount of material for an extremely limited excavation. But in this case, to tell the story of this particular collection, we've done everything from extremely detailed ceramic analyses where we're able to actually pinpoint not only who's making at the actual pottery where they're making these ceramic materials in the Staffordshire region of the UK, but the year that they were produced, and to archival research in the Danish West Indian archives, where I was working with an undergraduate student who also has a presentation in research dialogues to translate 18th and 19th century handwritten Gothic, Dutch, Danish, French, and Spanish to understand the property records of this particular excavated collection. So, and on top of that, we're drawing it in GIS and trying to understand kind of this very long-term 3-year field project, which just feels immense. So kind of to Catherine's point, sometimes it is very nice just to sit down and finish one part of it. But it does feel like an awful lot. So we're really at the beginning stages, but I think this is going to be a remarkable project at the end of the day. It'll kind of provide a brand new glimpse into what was for sale and what people were doing in this historic town.
Do you ever find that when you get into a new area of a type of analysis that it kind of can, change your whole understanding of some of the analysis that you've done earlier?
Yes, most archaeologists in this case would look at what we found and would just call it, blue on white transfer printed ceramics. And instead of a three or four word descriptor, we have gone kind of way down into the rabbit hole I mean, deeper into the rabbit hole than I ever knew existed with regard to historic ceramics. So yes, it's often surprising, and you know, and as Austin and Jacob kind of mentioned, sometimes you find materials that don't fit, and explaining those in the context of your research question can be pretty challenging. In this case, I've been kind of working with a hypothesis that all of my materials were generated during one New Year's Eve town fire on the cusp of 1832. You know, so I've been trying to kind of reject my hypothesis by dating all of the materials. So anyway, it is a constant interrogation of the data, I think.
Yeah. Some of y'all in the sciences, what has been most surprising about the work for you?
Sure. So Taya and I, both of us had never worked in a genetics lab before, and we weren't really familiar with the process of extracting DNA. And it was kind of intimidating at first because it is a lot of work, but it did get pretty easy with practice. And we were able to commit it to memory for the most part. And we didn't realize these skills are so versatile and that we could use them in so many different projects. So that was really nice to find out.
For me, I think the degree of precision that was required for my project was surprising, just because in every chemistry class you take, you know, a chemistry major or a chemistry professor is going to say you have to be very precise in chemistry. It's not like biology, but that is not true. If anything was even just like half of a microliter off, everything would be ruined. So that was definitely surprising for me. I see them shaking their heads. You guys can definitely relate to that. So that was very surprising.
Kristen, do you want to go ahead and ask the next question?
Sure. So our next question, knowing that you guys are all in different stages and have completed some research projects, what advice would you give to someone who's just getting started in research, or maybe just wants to get started in research and doesn't know what that would look like? So I know we've got a mixture in here too of some of you who maybe were more volunteered and assigned a topic, and some of you who maybe came up with something for personal reasons and wanted to explore. I know Vaidehi, you mentioned kind of based on your diet, you started wanting to look at different options. So maybe you guys can speak to that. And what advice would you give someone to get started in research?
Yeah, so for me, I think the biggest thing I would say is find something that you're interested in. Like I love cooking and making food, so that made my project a little bit more easier for me than if I did something with water salination or something like that. And also, if you ever get stuck, just do something. Just start. Like if you don't know what to do, just start. Once you start, then you'll eventually move forward and you'll figure it out. But don't just stay where you are because you're confused or scared on what to do next.
Yeah, going off of that, I would say even if you're outside of your comfort zone, keep going. I'm an English major and I was working on like an archaeology project. So when I was assigned it, I was a little intimidated at first. I was like, oh my gosh, I don't really know much about archaeology at all. And at first I was very outside of my comfort zone, but the deeper you dig, the easier it'll get. So just keep going, even if you feel a little scared about it.
Building off of what Taylor said, I was assigned my project and it seemed like every step of it was way over my head. I felt like it was kind of like imposter syndrome. Like I should not be on this project because I don't even know what this means. But it was great because Dr. Giles had done a lot of research very similar to this. So I was able to look at his past papers. And as I continued with my classes, particularly microbiology, I took that after I started our research. I started being able to connect the dots between what we were doing and my classwork. So that was extremely helpful. But I think the biggest piece of advice that I would have would be to look at past research, just of, you know, similar things to what you're doing, even if it's just somebody had the same or similar method section. So you'll at least understand the procedural part of it.
I would love to add just a tiny point to that also, because I so wholeheartedly agree with everything that's been said there. And also, this may have to do with kind of my learning style. I'm very reflection oriented. But for me, like, take a deep breath, sit down with a cup of coffee, and like write or sketch out some of your thoughts about things. That was a really great like practical way for me to get started. At one point I covered a table in like, I had one of those big like huge rolls of like easel paper, and I had taped it to a table and it was covered in sticky notes, and I just drew out like all the things I was thinking about. And this was when I had really very little idea of what the project would actually look like. Just because mapping out some of those ideas and getting them on paper really helped. But similarly, just talking to people, like not even just when you don't know what you're doing, but especially when you don't know what you're doing. And it doesn't have to be people who you think will know what you're doing. Like I talked to my brother and his fiance a lot, and it ended up being encouraging just to go back to the fact that she had said, she was like, that sounds fascinating. Like, I would love to participate in a research project like that. And it helped me realize, like, oh, this is not like people are not going to dread jumping into this project. This is actually, interesting to more than just me. So definitely would recommend taking those thoughts that you do reflect on and putting them out there, whether it's on paper or talking to people, just because it really can be surprisingly helpful.
So yeah, I definitely agree with what everyone said. It's very important to, look into the work of your mentor or, someone whose lab you're interested in working in, like reading their work and seeing what they've been working on recently, and just making sure that you're actually interested in the subject you're going to be working on. But sometimes maybe more importantly than that, it's important that you find a lab and a mentor that you truly feel comfortable around and that you can build a good rapport with. And just finding someone that encourages you professionally and gives you solid advice for moving forward in STEM, like career-wise, your next step in education. It can be really intimidating, but finding the right environment and practicing makes it a lot easier and actually makes science fun.
Yeah.
Yeah, I'd wholeheartedly agree with that. Jacob and I feel have been very fortunate with Dr. Smith and even Dr. Persons when he wasn't around, just working with us in the lab and kind of going over everything. And that would be my biggest point as far as getting started, even if you don't know anything about what you want to do. And you're kind of afraid of finding something or falling into something that you might not be as comfortable with. Reaching out to professors as an undergrad is so easy, and it's so much more diverse than when you get into more graduate levels, because you can just about go to anybody at the undergraduate level and get involved in their research. When you start getting into like a master's level dissertation, you're often working under a single instructor or advisor or maybe a small group, but you don't have as much freedom as you would at the undergraduate level to do the things that you want.
I think that's been great advice from our students of just asking, and any of the faculty on this call or researchers, like we love to talk about our research, so just ask, right? Dr. Pipes, Dr. Persons, from a faculty perspective, what do you have to add on advice for students interested in research?
I think the students pretty much said it all, but the one thing I heard that really stood out to me, I think Vaidehi mentioned, is just make sure it's something you're really interested in and do it. It can be anything, can evolve into a research project. If you have some sort of interest in it, you can find a way to make it viable research and to make it useful for society in some way as well. So just start with something you care about in some way. I think the most important part.
Yeah, I think everybody's done a remarkable job of following their passions and coming to this point. The best advice I ever got in graduate school, when I was in, I think maybe in a master's program, and I was terrified to figure out how to carve out a career and how to carve a niche out when, at least in my discipline, everybody kind of changes their theoretical orientation and, new ideas, like every field, right? New ideas come in and they challenge old ones, and then everybody's challenged to kind of reorient their research in different ways. And the best advice he gave me was to follow follow my passion and to follow the things that I was interested in and to continue to do good work, right? Not to worry about the kind of temporary fads and research as you get pulled in one direction or another, but just to do what you do and do good work. And then eventually it will work out. And it's hard to have faith in that, I think. But as you all have said, it works out, right? So I think y'all had great advice.
Yeah, this next question kind of builds on what both of our faculty members said, which is, by doing research, you're entering into a larger conversation, and the goals of research are really less than, getting a grade on the paper. They're about improving society. So the question is, what is the impact of your research?
I think,
sorry,
go ahead, Elizabeth.
Okay. For me, our antimicrobial peptides, that's what we were working on. They were discovered around the same time as antibiotics. And because antibiotics worked really well, really fast, there wasn't a whole lot of further research on these peptides. But now that we're moving into an era of antibiotic resistance and it's continuing to get worse and we aren't really keeping up with it, there's an extreme need to find an alternative. So that's really what we're thinking that these antimicrobial peptides could be used for. And additionally, we're looking at like targeting pathogens. So maybe eventually way down the road, they could be used as cancer treatment if we could just target, like radio label the cancer cells. So it's kind of ambitious, but we're in the starting parts of this.
And I'll kind of touch in here, I think, on the other end of the spectrum from maybe the more physical aspects of just meaning to the community in general, or what it means for the community. Our research really focused on preserving culture and history. Like Jacob mentioned earlier, a lot of the artifacts that we found range such a large period of time that each of those were connected to somebody in some way and tell their own story. And by collecting those and preserving them, which they'll end up getting sent to SEAC for the National Park Service, you really saved somebody's story in that, and it gives meaning to the people that may still be alive and connected to them today, just like it would in any other culture or cultural aspect. And I'm sure Jacob has something he can add about, especially who it could impact.
I believe it impacted the National Park Service themselves a lot due to the confirmation of historical accounts. It was extremely interesting having them sent us a historical map of the 92nd Illinois troop, or that regiment moving through at exactly the place that it says that they moved through. They dropped several Spencer rounds. It's one of the very first repeating action rifles of the time, and that was one of the only regiments that moved through, other than Wilder's regiment, that moved through the fields in Chickamauga. So it was the confirmation of the historical accounts. Really, I'm sure it made the Park Service feel a lot more comfortable in the sense of telling future visitors this down the road.
I would love to kind of add a quick note, because Vaidehi, your research stuck out to me because I'm not entirely vegan, but I do have food allergies and I can't have any dairy. And so I was kind of thinking about this general theme about how sometimes if you zoom in really close to a project, it may look like it doesn't connect to as much as it actually does. Sometimes you have to like back out to the 30,000 foot view because there may be like a few steps in the process. So while you mentioned vegan specifically, anytime that I can find vegan food, it's generally safe for me. And so that's something that I also really, really appreciate just from the food allergy side. With our project, it probably most immediately would have implications on testing second language acquisition. But one of the things that, as I was kind of just sitting and reflecting about it, I realized was that may have a lot of public policy implications. If you think about particularly kids in the US who don't, or or really anywhere, but I was thinking specifically in the context of the US who don't speak English as a first language. If that test isn't accurate, if there's a confounding variable there with creativity, and we're effectively testing the wrong thing or part of the wrong thing, then that could have big implications. So I think it's just fascinating to kind of zoom out sometimes and see, even if maybe it doesn't connect directly to something, it connects to something that connects to something huge. Does that, that sounds so redundant, but I hope that makes sense.
Yeah, and Catherine, building off of that, when I first started the project, I didn't really think a lot of like bigger implications, but then I started to realize, okay, the next step would be to make it a cheaper product, and then you could hand it out to homeless shelters and stuff like that. And you don't realize like people there also have food allergies. So like in this product or in the little jar, it's gluten-free, it's vegan, so that means there's no lactose in it, so dairy free. And then the only allergen is nuts, but then we can also create an alternative with seeds and stuff so that there's no allergens at all, and so that it can cater to basically everyone.
Let's see, who haven't we heard from? Juliet and Taya?
Sure. So the findings that we saw from our PCR amplifications, we will be able to contribute to the existing knowledge on the species diversity in freshwater streams. And we could also apply these methods to other species as well. But especially in North America, there are hyperdiverse regions that definitely need more studying. And there's also a broad impact testing DNA rather than just observing morphological characteristics to classify species. So we'll be able to add to a lot of those previous classifications that were just based on physical characteristics.
I will mention that the same thing is happening in archaeology to look at the diversification in both plant and animals that have been that are domesticated species. And so, you know, by going back and actually extracting the ancient DNA of corn or whatever, right? It's really contributing to our body of knowledge. So I thought your research was really quite cool, all of it. But I thought that was neat that you could identify such clear geographic and species distributions. And I was kind of wondering what was driving that diversity in certain areas, because I thought some of the, I guess it looks like the areas that were overlapping in your map were like northern Arkansas and Missouri, which those are not the most diverse, or maybe I'm unfamiliar with them, but those are not what I think of as high biodiversity regions like the Tennessee or Alabama in some places. So
really interesting, because there were actually several glaciation events throughout evolutionary history that's like really kind of screwed up the perception of diversity amongst these fish. And it's cool whenever new technology like DNA sequencing has come out over the years. It's kind of like you can go back and look at old studies and see that things are actually more intricate or like more diverse than previously thought. So it was really fun to work on this project.
I think in my case with scanning these documents, the purpose of the project was to make these documents more accessible to people, especially right now in the height of the coronavirus pandemic. Travel has been really restricted, and researchers that need and want access to these documents, they can't get them because they're in hard copy here. And if they can't travel here, they can't access them. So I think definitely the implications of this project can be bigger because it could span worldwide, different countries for people who want to access these documents.
Taylor, after scanning so much and handling all of that data, does it change how you think about what we preserve in the digital record versus paper records? Or for everybody here, for example, who's starting on research projects where you're generating maps and notes and data, you know, you've just slogged through hours of other people's research projects. So how do you think differently about data now than you did before?
Yeah, well, I mean, as I was like scanning things, you know, it can get kind of mindless sometimes, like you just scan, click, but sometimes things would catch my eye and I would just become more interested. And when I first went into it, I wasn't really that into data at all. I didn't think about it much. I don't have a very like science geared brain or anything like that. But it makes me realize the push that there is for digitization now. Because I mean, so many things could go wrong. A building could burn down, and then you just lose all the documents that are there, unless they're digitized. So I definitely understand the need for it now.
It's so interesting to see you all, making these connections between, earlier studies and then what can potentially, policy decisions that can be coming out of your research. And everything that we do in a scholarly way does make an impact, even though sometimes it feels really small, but it can make a difference in the long run. Kristen, do you want to continue?
Yeah, that was really, I'll echo, that was neat to see so many seeing the impacts to the community or to just preserving and educating. It is really, really neat, and finding the impact of your research, because it is easy to get a little, I don't know, I guess, sad or thinking that, well, who's reading this? Where is it going? But yeah, every little bit makes a difference and can have, I loved how Katie said, maybe the impact is in some of the like domino effects of this helps us learn more about this topic, which really helps us to change a policy or a practice or a medication, whatever it is. So that was really neat. So in our last little bit of time, and feel free still to throw in questions with the Q&A box if you have any that have come up as we've been talking, but we'd love to hear you share about the next phases of your project and how you'd like to continue your research. What are you hoping? What types of questions do you hope to answer next? So we can go in any order if anyone wants to jump out on that one, or we can start calling through people.
For my project, we focused on Vibrio cholera. So I would like to test the effects of these piscidins on different pathogens. And Vibrio, we were looking at it as a fish pathogen, but maybe in the future, moving towards human pathogens to see if really this is even a possibility to use it as, an alternative for antibiotics.
I'll jump in because we're in kind of an interesting situation. If you saw my video, you would have seen that we haven't collected all of our data yet. So actually we haven't collected our data yet. So we presented on the here's the project we're doing, and we've set up all our methods and stuff, but we're still waiting on that final piece of approval before we actually start involving people. So first of all, it would be just to complete my project there. But going from there, I'm really looking forward to all of the sort of peripheral insights that come, maybe that's just my nerdy brain, but all of the things outside of just confirming or not confirming the hypothesis of whether or not there is a real relationship, a positive relationship there, but also looking at how does it work for people to engage with the tests? Are we going to run into hiccups there? What's it like to do this online? And what might it look like in person? Might it be worth kind of replicating in person later? And then looking at different age groups, perhaps, or certain demographics, or even playing with maybe some other languages and see if that stays the same. So I'm sure I'll get overwhelmed soon with all the options once I get past this next bit, but that is definitely exciting.
And I'll tack on to what Katie was saying about taking and looking this in terms of other demographics or other age groups. What I'd like to do is look at it along with other individual differences that we know affect language learning. So it looks like creativity and language learning are related, but then there's anxiety, and then there's motivation, and then there's willingness to communicate. So there are all these factors that we know influence a person's ability to learn and use a foreign language. But the sort of next step is to see how they all fit together and which ones are important and which ones we can do something about and which ones we just have to deal with as they are. So I'm looking forward to pulling it out in those directions as well.
I know for Jacob and I's project, a lot of the bulk is done in terms of our GIS database that we've created, but it's still missing a lot of metadata that we're working on filling and carrying into overall, hopefully a publication as we seek publishing. And that would kind of secure the spot, just as far as being available knowledge, because this is the first time since the early 2000s that any real analysis at the academic level has come out of, in particular, Chickamauga, Chattanooga National Military Park.
I think the next step in my project is organizing all of the documents into an interactive bibliography. Right now, I scanned over 5,000 documents and they're on a hard drive. So the next step is definitely just organizing them into an interactive bibliography so that when we send it out, people can just go straight to what they want. And all the documents are text searchable PDFs, which just makes research that much more efficient because you can just search directly what you're looking for instead of physically having to dig through documents. So yeah, just making those more accessible.
So for the next phases of our project, Juliet and I are going to continue working with Dr. Alda throughout the summer. And just like other people have said, we definitely have more data to include. So our posts are now sort of preliminary to like the bigger picture that we're trying to go after. So far we have about 1,047, I think, sequences in our tree right now, but we're going to go on and finish with the samples that we still have to do for the cytochrome B gene. And then once we have all of the cytochrome B genes, we're going to go through and pick out specific nuclear genes that could be of interest as well and include those in our analysis. We're also going to try to learn more about the ArcGIS and mapping technology so we can get some more robust maps with maybe like river basin or different drainage information that can be a lot more, add a lot more detail to our work. And ultimately, we hope to be able to publish a paper eventually.
Yeah.
It's really cool.
Really, again, all of this research is so inspiring. I have so many questions for each of you individually, so I'm going to try to follow the example set by Austin actually and comment on your presentation pages later because there's probably not enough time for me to ask all of them of everybody. But yeah, I think I have a book to write. I've got to get through all the material that's sitting in my lab. We're continuing the archival research and trying to understand property ownership and the legal status of the individual who owned all of these, who lived on site and who worked in these spaces so that we can kind of broadly understand shifting social dynamics in the 19th century as populations became increasingly free and former slaves or formerly enslaved people or free people, right, of kind of an emerging middle class in some ways. But a lot more work to be done for sure, so.
Neat. And Vaidehi, you are in the unique situation that your project was part of your high school science fair. So really, really neat to see that involvement in research in high school still. Do you have any ideas of how you'd like to continue as an undergraduate researcher or directions you'd like to go?
I'm not sure about like as an undergraduate researcher, but as far as this project, I've started selling like the product that I've made at like local farmers markets and things. And right now my cost of goods is about $2. And I have been like any leftover jars that I have taking it down to the local food bank. But I think the main goal would be to get the cost of goods down to like less than $1 so that I can donate more without it having to necessarily be leftover, and then just balancing the business aspect of it with the charitable aspect.
That is really, really cool to see how you're making an impact already. So we're excited. We need to follow up and hear all that you're doing in a couple of years as you get further along in your academic career too. Well, thank you guys so much for sharing. I know it is a unique opportunity to get to hear from so many different disciplines and think about all of the impact of the research we're doing. I remember a professor thinking about all of the knowledge in the world as these like little tiny Venn diagrams, and everybody is building this incredible expertise in these tiny areas. And the more we have our areas, the more we're learning about our world and so many things. So this is always encouraging to hear lots of different disciplines and the work you all are doing. So we don't have any other questions in our Q&A that have come up. Do any panelists have questions for each other that they'd like in our last couple of minutes before we wrap up?
I have a quick question for Elizabeth. You were saying that we have like antibiotics, and then what was the other thing called?
Antimicrobial peptides.
Yeah, so what's the difference between them, and why would, like, if we do have immunity to antibiotics, or like if they're not working, what would make the antipeptides be different and work against that?
So antimicrobial peptides are something that all of our bodies actually have, and they have several conformations, I guess, and several modes of action. So particularly the piscidins that I worked with, if they were metallated with copper versus not being metallated at all, their activity was completely different. So the idea is that they wouldn't have the problems of resistance from bacterial species because their forms are so broad. Bacteria become resistant to antibiotics by recognizing, I guess just recognizing that antibiotic from exposure that wasn't enough to inhibit the bacteria, and then they can pass that information on to other bacteria, of course. So by using antimicrobial peptides, I think that we would completely do away with the problem, because it wouldn't be the same thing every time. Does that make sense? Is that what you're asking?
That's very interesting. So like the peptides are much broader, so it's not as easy to recognize them every time.
Exactly, yeah.
I have a question for Taylor. So I wondered about you talked a little bit about the interactive bibliography and kind of improving accessibility. And one of the things just in looking at psych, and a lot of my interest is in accessibility and neurodiversity and stuff like that. So I've noticed that there's a pretty significant overlap sometimes between making things accessible and then actually increasing people's like desire to engage with those things and ability to be interested enough to engage sometimes. So I wondered if that's something that you've thought at all about. I don't mean to totally put you on the spot, but if that's something you've thought about in terms of like, what's kind of your goal with increasing accessibility? Is it just to kind of make life easier for a researcher, which as a researcher I appreciate, or is there kind of a part B there of maybe looking at how you might get more people engaged and promote that material? Does that make sense?
Yeah, that's a really good question. Honestly, I think Dr. Smith and I were mainly thinking of just kind of making it easier for researchers to access it. But that is a really good point, and it's definitely something I should look into, because I know for me, like going into it, I didn't have that kind of interest, but coming out of it now I do, and I've kind of grown and fostered this appreciation for it. So I'd really like to see that happen for other people too.
That's really cool. Thank you.
I was going to.
Oh, go ahead.
I was just going to ask Dr. Pipes and/or Dr. Persons a quick question. Obviously, all of us had mentors like yourselves that we were working under, and I know both of you do a lot of research with students. I was just wondering, as primary researchers in the field now, do you guys like find it very helpful for mentoring undergrad students and being involved with their research as opposed to just your own?
Well, I can go first. I think it's wonderful. I think I encourage everybody who can get involved in to follow what they want to research. So I'm exceedingly pleased to see student projects of such a high caliber, like what we're seeing here in this session. So again, kudos to everybody. Yes, I think it is helpful. Sometimes in talking with you all about the things that you all are interested in, for example, it might make me think about something differently, even if it's on a different, even in a totally different culture area or a different time period. And to be honest, I had no idea that you could date rifle casings the way that you all have done with some of your material culture. So I've actually learned something from your research as well. So yeah, I think it is. I think it is helpful.
I thoroughly agree on several levels. One, it keeps me excited about it because to have somebody new come in and see things for the first time and get to explain them to someone for the first time, it reminds me frequently of why I'm interested, why I got into this sort of research. And two, it keeps me current. That's the best thing about reading student projects is that you get new ideas and it helps you keep in touch with sort of the fundamentals of it and then new ways of thinking of things. So I certainly think it helps me a lot.
Thanks.
I think one of the things that is also great, as we've seen in this session, seeing the kind of connections between biology research and archaeology research way that, you know, specialties of each area can help inform the others, is that kind of collaboration is really key to making really leaps forward in our understanding. So it's been great to see that in this session.
So everyone, we are out of time, but before we really do run out of time, I want to thank everyone for participating in this session today. Thank you so much to our moderators. You guys did a fantastic job, and the presenters did a wonderful job. We're excited to revisit your presentations on Symposium. If you want to engage further with the audience this week, we invite you to do so through the comments tool in Symposium. I was just looking and we're already getting some of that in comments. So that's a tool for you to use. You can set up a time and day to actually be online and do some great chatting with folks through the comments tool. So we encourage you to do that. Thank you so much. Enjoy the rest of the week and the sessions here. We've got one more at 4:30 and you're certainly invited to that. And we look forward to seeing you soon. Awesome work. Great work. Thank you.
Thank you.
Thank you.
Thank you all.